Okay, now we're going to start the second part of our morning session, which is good sex. So we think of the translation of DNA and our chromosomes, they kind of dictate what we get from our parents, like our hair, nose, eyes, and even our behavior. Okay, I apologize. So our sex chromosomes dictate if we're male or female. But now we know that from the symposium, we know that there's a lot more going on, that the dogma is not so easily explained. So we know that our gene networks and the environment are involved, and they manifest in our sexual fate as well as our identity. We know that environment, society, and community can affect our health as well as health care when it comes to our sexual fate as well as identity. So our next two speakers are actually going to talk about sexual fate and identity. Our first speaker is Judith Kimble. She is a Vilas Professor of the University of Wisconsin-Madison and investigator of the Howard Hughes Medical Institute, and is elected to the National Academy of Sciences. Another product of the great research in Boulder, she received her Ph.D. from the University of Colorado, and she postdoced at the Medical Research Council Laboratory of Molecular Biology in Cambridge in the UK, before joining the Department of Biochemistry at University of Wisconsin in Madison. And the title of Judith's talk is The Ambiguity of Sexual Fate Determination. Judith?
You should wait to clap to hear after see if I'm worth it, okay? So is this too loud? It's okay? It's too loud for me. So I want to thank Larry and the other organizers for inviting me here. I had to think about what I wanted to talk about because I am a scientist that's different from most of the talks you've heard so far. I don't work on disease. I don't work on humans. I work on very basic science principles. And I wanted to talk about something that you might be interested in and also that Larry might be interested in. And so I picked this, which I've been working on actually for quite a long time, but I almost never talk about it. And I was here a couple years ago and gave a seminar, and I talked about the main thing that's going on in my lab, which is stem cell regulation. And I thought, okay, I'll talk about something different here. And I thought I would start by, let's see, talking a little bit about the fact that I was a graduate student here, not in Larry's lab. But as a graduate student, I learned a number of very important things. One is that we really don't know very much. This was true in 1973, and I think it's still true. And I think that that will come out of my talk. We know more now, but we still don't know a lot. That it's very important to focus on big unsolved problems, and that if you're going to tackle those big problems, it's really important to tackle them in an organism or in a system that is experimentally tractable. And of course, genetics is awesome, and if you can use genetics, that's a great thing. Okay, so when I started in 1973, I had my big unsolved problem that I was very interested in. And that is shown in the next slide, which is how okay, how does a fertilized egg generate a complex creature? Now, when I started in 1973, I didn't actually frame it in quite this way. But rather, I framed it in this way. This was probably the complex creature that I was most interested in. This is Eleanor Roosevelt, who was a very complex creature and really a remarkable woman. But once I started, I reframed my question to, and I still don't know the answer to this one. But today what I'd like to do is talk about a slightly different version of this, which is a different complex creature that I think all of us would really like to understand. Okay, so these were the kinds of questions I was very interested in. When I started as a graduate student, I'd actually been working for a couple of years in Copenhagen on human fetuses and trying to understand these questions in humans. And I realized this was just not possible. And I actually came to Boulder to work on coli because I wanted to understand molecular genetics and be able to approach my problem in that much more sophisticated way. So to tackle it with an experimental tractable organism, I was really lucky that when I started, there was an assistant professor in the department who was working on a small worm that was, in fact, what I consider the E. coli of animal development. So instead of working on E. coli, I started working on the worm because this worm is made of the same basic tissues that we are. It's got muscles and nerves. It's got a gut. It feeds, it poops, does everything that we do, but it has a 3-day life cycle. So that if you start an animal as a fertilized egg, it will have grown up to an adult by the time a yeast colony has been generated on a plate. So this is a very tractable organism. It's got 1,000 somatic cells and about 2,000 germ cells. So it's also very simple anatomically. You can freeze it, you can do genetics with it, and of course, over the years, the power has increased phenomenally. So I started my work in the laboratory of David Hirsch, who did a very brave thing, going from working on tRNA to starting his lab on worms because he thought that it was going to be important. And then I also was very influenced by Bill Wood, who joined David's lab as a sabbatical visitor for a year and was also a very big influence on me. So this was my graduate work, and what I want to do now is fast forward through my postdoc. I got my Ph.D. in '78. Fast forward through my postdoc and my first few years as an assistant professor to how I was focusing the question that I was most interested in at that time, which is how stem cells are regulated. So stem cells, you all know, self-renew and differentiate. And I had found as a postdoc, I'd been very lucky to collaborate with John White, who had developed a laser microbeam to kill individual cells. And in doing that, I had discovered that there is a cell that signals to the stem cells and maintains them. And so this was really the question that I set out to work on and learned cell signaling regulates stem cells. This is a concept that has stood the test of time: worms, flies, mice. And if you think about this very simply, you can think about signaling from a stem cell niche that has to be received in the stem cells. I spent probably the 80s and 90s working on the signaling part of it, and now I'm spending quite a lot of time trying to figure out how the stem cells can hear and decide to be totipotent. So these are very basic problems. And in this field, virtually everything that we've run across in worms has been true in principle and oftentimes in molecular detail in mammals and humans. Okay, so I haven't yet talked about the ambiguity of sex determination. How did I get there from working on stem cells? Well, it turns out that stem cells come in 2 flavors. They come in males and they come in females. And that the somatic cell in a female will drive a stem cell, and when that stem cell differentiates, it will differentiate in a female mode, whereas a male stem cell will differentiate in a male mode. I was working on this in germ cells, where it was a very simple male-female decision. which is to make an oocyte or a sperm. And we learned very quickly that signaling also regulates sexual fates in both of these sexes. And so now I'm going to leave stem cells and talk about how signaling controls sexual fate in both sexes. And this is, again, a concept that's been found in worms, flies, and mice. Okay, so I thought now I've sort of kind of taken you to how I got into this problem. I wanted to give us a sort of a basis of understanding the concept of sex determination. And I think it's already been mentioned that the primary signal is chromosomal. This is true in humans, where the primary signal is the XX versus XY chromosomal. But it's also essentially the same in worms, flies, and mice. Molecular details completely different. But conceptually, the chromosomes are the primary signal for sex determination. Now, also in all of these organisms, sex determination occurs very early. And so here we have a human fetus, which is at 7 weeks, and at 7 weeks has what is called a bipotential gonad. It doesn't know yet if it's a boy or a girl. Then by 8 weeks, that bipotential gonad has developed, because of the primary signal as chromosomes, as a testis or an ovary. And then the testis makes testosterone, which goes on to signal to the rest of the body and coordinate the sex of the male sex organs. Is this news to you, Larry? Oh my gosh. These are the basics. Okay. So if we think about the basics of sex determination then, we can think about them in this somewhat antiquated view, which is that it is bimodal. The primary signal is chromosomal. That signal is read very early during animal development. It's determining the sex very early, and the intercellular signaling coordinates sex across the entire organism. And these basics have been known for a long time, even if Larry hasn't known them. And lots of molecular specifics have been unraveled over the decades in this. And what we've been working on primarily is how this intercellular signaling coordinates the sexual fates and how they are read. So again, I'm going to give you this simple cartoon which says that cells talk to each other, cells signal, and those signals act throughout the organism. But then each cell has to read that signal and interpret that signal. Now, in the work that I've been doing, we've been interested in how that signaling drives the sperm or oocyte decision. And here, there is signaling throughout the organism, but it's being received in the germline tissue. And the question is, how is the germline tissue receiving that? And again, we've been working on this for, I hate to say it, but over 30 years. And we actually are slowly making progress in understanding this problem. So what we have unraveled is a tissue-specific molecular network that's drawn up here. The main thing you can get out of this is it's complicated, that there are master regulators of the sperm fate that we've been able to identify. If these regulate the program of spermatogenesis versus oogenesis. But those master regulators are controlled by cell signaling. This is a divergent hedgehog pathway that's controlling it in this organism. It's also controlled by a puff RNA network, and I don't want to go into what that is. It doesn't really matter. It's just molecularly different from the cell signaling pathway. And it's also being regulated by ERK-MAP kinase. And so these three are converging on the master regulators to determine whether or not that germ cell will differentiate as a boy or a girl. Now, it's not really a boy or girl. It's a sperm or an oocyte. But it's the sexual fate. Oh, I wanted to. So there's a paper that's just come out, and it's come out from MCDB, from Min Han's lab, that has shown that, that fatty acids from the gut actually impinge on this network. And they impinge on the MAP kinase part of it, but they are coming from the gut, and therefore that fatty acids and metabolism from the gut can also affect sexual fate. And if you take this one step further, which of course nobody has done yet, but just take it one step further, you could imagine that the microbiome and its production of different chemicals in the gut might also affect this network. Okay, so because we knew a lot of the molecular detail of how a germ cell is specified as a sperm or an oocyte, we could ask when that happens. Now, what I told you at the beginning was that sex determinant occurs early, and this is the dogma in the field. But now that we knew this in a specific tissue, what the key regulators are, we can take them out, we can put them back in, we can play with them, and we can ask, when is it regulated? And we decided to start these games in the adult because there was no way they were going to work there, and then we could work backwards. Okay, so in the adult, there's a female cell that's signaling to a stem cell, and that makes an oocyte. When we then genetically either added a sperm fate specifier or genetically removed an oocyte fate specifier, what we found was that in that same adult, we could sexually transform the tissue. We did not expect this. We then went on to males. Can we do the same thing in males? We genetically added oocyte fate specifiers there, or removed sperm fate specifiers. And again, we could sexually transform the tissue. Now, I want to note that the first time that we published this was in 1987. So we've known this for a long time. We did this first with one gene that we knew. Then we've done it with multiple genes. Other labs have done it. We all come to the same conclusion, which is that we can switch this germline tissue from oocyte to sperm, sperm to oocyte, as an adult. Not anything we had ever expected. Now, one might ask, so this just summarizes this. We're altering key regulators and doing sexual transformation, but always, this is not the whole organism. These organisms still have, a female still has a vulva, a male still has its worm equivalent of a penis. That's still, it's with the internal workings that are being transformed. Now, what about other organisms? Does this happen in any other organisms? Well, I think it's been known for a long time, for many, many, many decades, that there are certain fish that can switch from one gender to another as an adult. And this was always considered very weird and a total mystery, and I think people are still not working on it in any depth. I don't know, I've done Google searches. I don't really understand the molecular basis of this yet, and I don't think people do. But it's something that's been known for a long time. Okay, so what about other organisms? What about flies and mice? Does this happen in them as well? Well, it was not known for a long time, but the key here is identifying the key fate regulators so you can then manipulate them. And what's been found first in mice and then more recently in flies, but in mice it was only in 2009 that the first case of this was. And this is somatic cells within an ovary are transformed to male somatic cells. So you have, if you take out the key transcription factor that's saying be female, it will now start making the male program. And similarly, if you take out key regulators from a testis, and it's not that the testis as a whole is being transformed into an ovary, it's that key cells within the testis are being sexually transformed. And this is also seen in flies. So I would argue, anyway, that if we think about the basics of sex determination, that we have to add one now to that. And that is that sexual fates, at least in individual tissues, can switch in adults. Okay? And that this has been seen in worms, in flies, and in mice. It has not been shown in humans, to my knowledge, but I would be very stunned if it were not true in humans as well. Okay, so instead of sex being determined early, we would say now it begins early. Sex determination begins early, but it's a continuing process, and it must be maintained. Okay, so let's go back to worm germ cells for a minute, because there's one other thing I want to tell you that I think is totally mind-blowing. And that is what I call sex transformation with a twist. There are certain mutants. So remember, we have this elaborate pathway now, and we have a lot of abilities to manipulate that pathway. And there are certain mutants where if you start a female that's making sperm, you can add a chemical to that adult female and switch her into oogenesis. You can start, this is all of the females, 100% of the females are making sperm. You add the chemical, 100% are making oocytes, and those animals then are fertile. So these gametes actually work. So this is kind of amazing, adding a chemical. Now, one might say, so what is this chemical you added? We know enough about the pathway to know what chemical to use. I can tell anybody who's interested at the end if they want to know. But basically, we knew what inhibitor of ERK MAP kinase signaling we could add to do this. And multiple different inhibitors of ERK do this. Okay, so what we're saying now is that not only can we take out a gene and cause sexual transformation in adults, now we can add a chemical and cause sexual transformation in adults. Now, I show up here, this is in certain mutants. Now, in certain mutants this is true, but in other mutants it's not true. And yet, these mutants all look the same. And so, a cell that has adopted a male fate, in some situations, some genetic contexts, are different from a cell that's adopted a male fate in another context. Is it getting too complicated here? Okay, so we have chemically switchable and not chemically switchable genetic variants. Now, we wanted to know what the difference is. What's the molecular difference? And Elena Sorokin joined the lab. Elena had a really interesting history. She was a history major at Harvard, then went off for a couple years to learn biochemistry, and then came to my lab because she had become infected with omics. And we had been doing some omics, but she really wanted to do omics. And so we decided we were going to do some omics on this problem. Maybe that's why I got invited. Anyway, so what she wanted to do was take these certain mutants that can switch and compare them to the mutants that cannot switch and compare their transcriptomes. Now remember, these are comparisons between animals that are identical morphologically. Because the germline mass is 2/3 of the volume and mass, probably more of the mass than of the animal, we can just do the whole animal. And we can grow these up in 10 to the 6th in a liter, because these are like the E. coli of animal development. So we can compare these transcriptomes. And this is what she found, is that, that if she compares the transcription, she actually detected lots and lots of RNAs, 16,000, and most of those were identical in the two mutants. They were identical based on significance criteria that we decided to make very stringent because we really wanted to believe the things that weren't identical, and everybody who does omics knows you can get a list to satisfy any hypothesis that you're interested in. I shouldn't say that too loud here, I think. Anyway, so we used an FDR cutoff of 0.01, and we demanded a twofold cutoff so that we could believe these. And what we found that only 135 went up and 84 went down. I think that's a gross underestimate, but I think we can believe those. And the 135 were very strongly enriched for oogenic RNAs, even though these were generated from animals that were making only sperm, whereas the 84 down were not enriched in any way. Elena went on then to do what's known as principal component analysis to try and separate out and organize this data. And what she found was that there were intersexual RNA signatures in these animals. So if this is the normal female and this is the normal male, the ones that could not be switched chemically were much more similar to a male than those that could be chemically switchable, even though they look the same. She also looked at proteins. We did not do omics for the proteins. That was too hard. But we looked at individual proteins that we knew were male-specific or female-specific, and we found that even some of the proteins that were female-specific were coming in these spermatogenic germlines, but they were coming on early and then shut off again. So clearly they were being expressed, the RNAs were being expressed, they were being translated, but then they were being shut down. So remember, these are all of these situations have a male fate, at least in the germline, and we're comparing them to oocytes. And so if we stand back and think about this, what we're seeing is that, at least for worm germ cells, we can get a sexual fate that looks male, but if you look inside of those cells, they are different, and that they have a spectrum of molecular signatures. And this is, I think, a different way of thinking about things than people have been looking at them before. Now, I started these basics of sex determination, and I'd like to add one that I think is going to probably be true, but we only know so far for worm germ cells. And that is that there can be molecular intersexuality in cells and tissues, at least in certain genetic variants and with certain chemicals, right? So this can exist, not something we knew about. Now, when I was a graduate student, Larry many times said, one of my favorite pastimes is, quote unquote, thinking off. I'll leave it to you to understand what that means, but it's not hard. So I think what I'd like to do, because this is a gold symposium, I thought I would take what we know and just extend it, because that's the fun of it. It's fun taking the basic principles that we're understanding in something as weird as a worm germ cell and think about it in broader context. So this is what I would suggest we're addressing here. We have known for a long time that gender is bimodal, but I would argue that this is an old-fashioned way of thinking about it, and that this is a very puritanical way of thinking about it that has been designed by our Western society. And we now, I think, are in an age where we're starting to question that. And this says beyond he or she. It was just on Time magazine's cover. So this is very clearly something that's in the public eye right now. So I would say that we have shown that gender is not bimodal, at least at the molecular level in worm germ cells. And so if we then take that idea that there might be a spectrum of molecular signatures, we can now apply that to a human. Remember, this is the quintessential thinking off. I have no data for this whatsoever. However, we could think that this individual might have a spectrum of molecular signatures in some of his or her cells. We are not missing pronouns here that are appropriate. And that this could even be expanded into neural circuitry. So far I've said that a cell could be intersexual individually, molecularly, but what if a circuit was intersexual? What if a neural circuit or a neuroendocrine circuit were intersexual? What would that look like? And one can, of course, then, we haven't shown it for the female situation. We've only shown it in male, but this can also be true for females. There's no reason not to extend the reasoning. So so is this a reasonable idea? One of the things that we have been able to use is these genetic variants that we designed in worms. However, I think we all know that human genetic diversity is vast, and within that human genetic diversity that is vast, it's very likely that there's some people that might have the equivalent, and it would obviously not be the same, but the equivalent genetic diversity that we've been talking about in worm germ cells. Worm human genomes change, so you could imagine very easily that somatic mutations might arise that could cause this kind of thing. Again, no data, no evidence. This is just sort of taking it the next step. And then if we think about the chemicals in our environment, I think it's pretty clear that we are all being deluged with chemicals in our environment all the time. So I'm left with the question, can genetic diversity or chemical pollution or differences in microbiomes, I think is another one, could they influence sexual fate in cells, tissues, or circuits in humans? This is something we have no idea if this is true. I think there's lots of potential for that. And I would actually argue that it's important to try and refine the question a bit and ask whether or not genetic diversity or chemical manipulation might change the sexual fate of neural circuits or neuroendocrine circuits. Now, you could ask this in humans, but that's hard. And remember, when I was a beginning graduate student, I learned it was important to work on problems in an organism or system where you could actually get an answer. And so I would argue that this question is best worked on in worms and flies, even though some people would say there's too much research on these organisms with little relevance to humans. So I'd like to acknowledge the people who've been working with me over decades now. My initial people, Kathy Barton, worked with me in the mid-'80s, Tim Shadle, Ron Ellis in the 90s to work on plasticity of adult sexual fate. A MD, Ph.D. student in the lab worked on chemical switching and really discovered the fact that you could add a chemical, and within 15 minutes of adding this chemical, you could get a switch at the molecular level, and within 2 hours, you could start seeing differences in the sexual fate regulators being expressed. And then Elena, who did a great job identifying these molecular signatures, and we worked in collaboration with Audrey Gash, who's a real omics person. And I'll take any questions. Thank you.
This is to your last point, Judith. So everybody knows that when people become adolescents, they have a burst of hormones, and then they become interested in the opposite sex or whatever. But not too much, I think, is known about self-identity. In other words, you hear these people who are transsexuals who say, often, I felt like I was in the wrong body. I feel female, I'm in a male body, or vice versa. So, and I don't think anyone knows what the endocrinology is behind that. So what have you seen in neuro signatures with your gene expression studies, if anything, that you think could help answer that question?
So we have seen 0 in terms of the neural signature for many reasons, in part because we are manipulating the genes that are specifically controlling germ cells, and we wouldn't expect to see that. However, there's a lot of work being done in worms on the neural circuitry of males versus females, and there are genes that are known that regulate that. They haven't been removed in adults to ask if you can switch, and I think that's a really interesting possibility that's experimentally feasible now to do. But so I actually think that we are mosaics in some way, and yet I think it's going to be really complicated. If you think that my little genetic circuit is complicated, and it can be influenced by chemicals from the outside, chemicals from the inside, humans are going to be much more complicated. And going in and trying to, I'd like to, over the next 5 years, understand this in worms and flies, see if it's true at a neural level. We don't know that that's true. We do know that the brain is very plastic. I think that's something that we all understand. So my guess is that the brain will also be plastic with respect to sexual wiring, but I don't know that. That's a guess.
Judith, enjoyed your presentation. Can you, in this context, address the use of aromatase inhibitors, which in birds have been used to switch?
Wait, sorry.
Aromatase inhibitors.
I didn't understand.
Aromatase?
Aromatase.
Yeah. And whether there are any evidence of somatic mutations there. Definitely people have used inhibitors to switch the gender and the behavior and the neurochemistry of birds.
Right.
And what's known in mammals.
That has no relevance here. It's the principle that molecular, yeah.
Thank you. That was fascinating. And I just wanted to put a quick plug in for a book that's for sale in the lobby that I read a few months ago called The Gene, which has a pretty extensive discussion of some of the mouse experimentation in this area.
So some of the people in the audience may know that C. elegans are hermaphrodites. So when you say that you're changing a male into making oocytes, I assume you mean a true male that's just male? So then can they self-fertilize at that point? Because they should have made oocytes and then sperm.
A hermaphrodite can self-fertilize.
But inside the male, even though they previously made oocytes and now are making sperm, those can't.
Yeah,
Okay.
they can't support it. And I sort of skim over that because as a adult, they really are females. They've made some sperm early on. And a very closely related species is male-female. So being able to twiddle the circuitry is a fairly recent evolutionary phenomenon.
I have two things. One, I have a transgender, my oldest child is transgender, so I'm very interested in the topic. But at the Boulder TEDx this past fall, there was a young woman who is intersex, and I had not heard of this before. She did her talk on it, and she appears female, but she also has a Y chromosome. And she talked about the fact that there's about 1% of the population in this category that most of the time when they're babies, surgeries or things are done, and so they kind of are underground from a research perspective. So I'm just wondering, when you talk about not doing research in humans, if there's anybody doing work on intersex population.
So it's been known for a very, very long time that there are intersexual people and animals, not that people are not animals, but those are usually identified by genitalia, differences in genitalia, and so they're very early events. And so that's been known for a long time. I think what hasn't been known is that the internal workings might also have this intersexuality. And I think that's very important for our conversation today, and I hope I've set you up well.
Okay, one more question.
Just two quick things. First of all, in terms of chemicals, I remember when I was living in England, we had a big issue with estrogen in the waterways and fish changing their sex spontaneously. But I was really intrigued by this, linked with all the other talks and other things we've done in past presentations. We have epigenetics, and we find that suddenly that the DNA, the expression of DNA, can actually change within me, my generation.
Absolutely.
We have the microbiome, and now we find that the network signaling may be affected by outside influences such as an infection and can cause problems. Now we're talking about really fundamental switches caused at the stem cell level and in the signaling. All of this in relation really to changes in contexts in which those cells operate. And so a question I throw out in terms of this broad Larry thinking, but it has concerned me for a long time, is has anybody actually thought about or looked at, I'm sure they've thought about, the impact on the human beings of climatic change as we go forward? Because we are extraordinarily sensitive, far more sensitive to context than we had realized. And I just throw that out as something to think about, because this is, the complications.
I think climate change is going to affect all of us in many ways, including perhaps our sexuality, but I think in more profound ways like that, like drowning if we're living in Florida.
Okay, we'll take one last question. I'm sorry.
So in your principal component analysis, you found some genes which are not switchable at the bottom where the fate is already driven. Have you thought about, are there chemicals, or can you completely switch them? There may be some association that even if they are not switchable, but have you tried to see?
And so that was done on animals that had not been seen a chemical. So that was done on the animals before chemical treatment. We also did a whole lot on after treatment, and we didn't find anything that we thought was that interesting or surprising. Their oogenic program got turned on, and we could figure out the kinetics. But what was surprising to us was that looking at these two different genetic variants without the chemical, that they were molecularly distinct and had an intersexual RNA signature. So does that make sense? And we could talk more at the break. I should probably.
Thank you, Judith.